Okay, so the topic today is the small intestine, and let's just go into it here. So, the small intestine, its main functions, starting off. The small intestine is where the vast majority of chemical digestion and nutrient absorption takes place. People think of the stomach as the main digestive organ. The stomach actually plays a relatively minor digestive role. The acids begin to break down polymers, but without enzymes, it isn't really sped up all that fast. The pepsin, which is present in stomach juices, is, going to make a rough chop on proteins, but it's not what's really going to chop proteins down to their individual amino acids. And almost no absorption happens in the stomach or esophagus. The small intestine is the largest, or at least longest, part of the digestive system. We call it the small intestine. That's because of its diameter, which is smaller than the large intestine, not because of its length. It's much longer than the large intestine. It's about 5 meters long in a living person, about 8 meters long in a cadaver, and this is because in a living person you're going to have some muscle tone and keep those smooth muscles somewhat contracted, which tends to shorten it. There are 3 divisions of the small intestine: the duodenum, the jejunum, and the ileum, in that order. The duodenum is the first section of the small intestine. It links to the pyloric sphincter. So it's the first thing that food enters when it exits the stomach. It curves around the pancreas, because the pancreas is going to rest in this area in here. Transverses the abdomen and then starts to make another curve where it will go back. You can't exactly see it here because it's obscured by the large intestine. That is the duodenum. It's just this basically first sigmoidal section, curve coming off of the stomach, going laterally, and then curving again. Once you get past that point, you're out of the duodenum. The duodenum is mostly going to be known for its interactions with the gallbladder and pancreas. As you can see here, this is starting at the pyloric sphincter, and it ends here at what's called the duodenojejunal flexure. That's that last bend. The duodenum is where chyme enters from the stomach and first begins to be mixed with pancreatic secretions. All right, most of the digestive enzymes and other substances that work in the intestines come from, or at least through, the pancreas. And so right here, you're going to have, that's where the pancreatic duct is, and you're going to have pancreatic juice coming, as well as bile, because you'll have bile coming down from the gallbladder through the pancreas and also being spilled into this area. The functions of pancreatic juice, the specific functions, we'll get into a little bit more depth when we cover the pancreas, but pancreatic juice contains basically two things. One is a basic buffer, mostly sodium bicarbonate. And the purpose of that is to neutralize the stomach acid. The stomach is protected from damage from acid by mucus and other things. The small intestine is not, or at least most of the small intestine is not. So the first thing that you got to do is neutralize the acid so you don't end up burning and ulcerating your intestines. Secondly, the pancreatic enzymes digest starches and proteins and some lipids as well. The function of bile, at least from a digestive perspective, is to break up fats. Fats are not actually susceptible to digestion until they've been dissolved, and bile salts basically act as a detergent that are going to solubilize the fats. Pepsin is going to be inactivated. Pepsin is that protease, protein digesting enzyme from the stomach. It's only active at low pHs, so it's going to be turned off. And pancreatic enzymes will take over as the main digestive enzymes. There are also some digestive enzymes that are basically made by the intestinal cells, and they operate only very, very close to the edges of the intestines. So that is the duodenum. The duodenum is where, so keep this in mind, this is where pancreatic enzymes, pancreatic buffer, and bile salts are going to be added to the chyme, and movement through the duodenum is going to mix the chyme with those secretions. The next section of the small intestine is the jejunum. Once you're past that duodenojejunal flexure, the next 40% of the small intestine, about 1 to 1.7 meters in a live person, is the jejunum. And you can see it here, this area that is highlighted in purple maroonish. That is the jejunum. The jejunum is where most digestion and absorption occurs. So remember, the digestive juices, the enzymes, were injected in the duodenum, but food is going to move out of the duodenum before those enzymes actually have enough time to get to work. Where they're going to be working, doing the digestion, actually breaking apart all of that food, is in the jejunum. And as that food is broken down, it is absorbed by the epithelium of the jejunum. There's a reason why this picture has the jejunum in that sort of red-maroon color. That's because the jejunum has a lot of blood supply. It's very highly permeated with capillaries, and that makes sense, right? Those capillaries are there to take away the nutrients. This is where nutrients are going to be released by digestion and where they're going to be absorbed into the blood. Pretty much everything that is relatively easily digestible is going to be digested in and absorbed in the jejunum. It has circular folds, these large, tall, closely spaced circular folds that look like ribs, kind of, spaced throughout it. Those increase surface area. It has a relatively muscular and thick wall. That allows the jejunum to undergo a lot of churning. It's going to keep mixing so that, remember, things are only absorbed when they come in contact with the wall of the intestines. So you have to keep mixing the chyme as it moves through so that nutrients that have been digested towards the center of the chyme can be moved towards the edge where they can be absorbed. So you continue to have a lot of mechanical mixing all throughout the jejunum. The ileum, which you see here, is the rest of the small intestine. It's the second 60% after the duodenum. It's thinner, less muscular, less vascular, and more of a pale pink color when seen in the body. Most of the nutrients have already been digested and absorbed by the time chyme gets to the ileum. That means that for the most part, stuff that is in the ileum is going to be stuff that passes out of the body, that will eventually become feces. There are some particularly difficult to digest elements that will be absorbed in the jejunum. Not a lot of them, but there will be some. The ileum also contains a special lymph node called Peyer's patches, or Peyer's patches. These are lymphatic tissue clustered throughout the ileum, which are going to help protect you from any contaminants, particularly pathogenic contaminants, present in the food that you ate. And before that food gets to the small or large intestine. The ileum ends at the ileocecal junction. That is this sphincter right there, the ileocecal valve. And you'll note that it's spaced a little bit up. It's not right at the end of the small intestine, or at the large intestine. You actually have this little loop below it, and then this little hangy down thing there. That's the appendix. Well, food will come in and fill this area down here. That's the cecum, before it moves on through the rest of the large intestine. Basically, what happens is that the valve remains open until the cecum is full, and the valve closes off. And then once this food, or chyme, or pre-fecal matter, gets moved forwards a bit, then the cecum is empty again, and the valve can open up, and more matter can pass in. So that's the gross anatomy of the small intestines. Let's take a look at what it looks like on the cellular level. Now remember, the primary function of the small intestine is digestion and absorption. And a lot of it is optimized for that absorption function. What is going to increase the rate of absorption, your ability to absorb things? Surface area Absorption only happens at the surface. So everything about the small intestines is intended to maximize surface area. That's why it's so long and twisty. The lumen contains simple columnar epithelium. The fact that it's simple makes it much easier to move absorbed nutrients through it and into the blood supply. But it is columnar, making it a little bit tougher and also giving plenty of space for tight junctions. It's important that you would find tight junctions all throughout the small intestine because you have lots of digestive enzymes in there. You do not want those enzymes getting loose in the rest of your body where they would digest the rest of your body. There are 3 types of internal folds that increase surface area of the small intestine. First off, the circular folds. These are those circular ridges that are particularly present in the jejunum. They're present throughout the intestine, but they are particularly pronounced and abundant in the jejunum. And so those are the plicae circularis, or circular folds. They increase the surface area by about 2 to 3 times. On the circular folds, so you would have, if you're looking at a longitudinal section, you would have this as a circular fold, and then down, and then another circular fold. Well, if you zoom in on this area here, what you will see is that even in that small area there are a bunch of villi, projections going back and forth. So each of these circular folds has a bunch of little fuzzy finger things coming off of it. These increase the surface area by about a factor of 10. We're not done yet, because if you were to take a look at the villi, magnify that out, you would see that it's got columnar epithelium here, and you have microvilli projections, what's called a brush border, at the top of each of these cells. So even the individual cells have little projections coming out to increase surface area. Those are going to increase surface area by about a factor of 20. The circular folds themselves are only folds in the mucosa and submucosa, and they occur from the duodenum to the middle of the ileum. They're particularly abundant in the jejunum, fairly small and sparse in the ileum. That's because most nutrient absorption happens in the jejunum. They do a couple of things. They increase surface area. They also disrupt the flow of chyme, right? So as chyme is flowing through this area, eddies are going to be created as it passes through the circular area here. That will cause the chyme to sort of automatically whirl and mix. As the chyme mixes, that's going to bring digested nutrients from the middle of the chyme in contact with the surface area of the intestine where they can be absorbed. Villi, which you can see here, are finger-like projections found on, well, pretty much any epithelium in the intestine, but they're going to go up and down the circular ridges as well. So you can see here there's a bunch of cells, simple columnar epithelium. These are about 1 millimeter tall, and they give the small intestine a fuzzy appearance. There are 2 types of cells that you'll find here. Absorptive cells, or enterocytes. Entero means inside. So enterocytes bring things inside. That's most of the cells. And you're going to find goblet cells sort of interspersed throughout. Those secrete mucus. That mucus helps to protect the lining of the intestine from digestive enzymes. And these epithelial cells are going to be joined together with tight junctions, of course, as I said. There are basically inside of the villi, you find lamina propria material, and each lamina propria material inside of a single villi will typically contain 1 venule, 1 arteriole, and 1 lymphatic ductule, called a lacteal. And then, of course, the arteriole and venule branch out into capillaries, which are going to be where absorption takes place. Microvilli. Now, if we take a look here, and sort of zoom in, you can see these coming out from an individual cell, these little projections here. And this picture doesn't really give it justice, because it looks like there's like 5 or 6 of them on one cell, when in reality there would be thousands, perhaps tens of thousands of microvilli on each cell. These serve to increase the surface area even more. In fact, I would say that they probably are the thing that increases the surface area the most. They're at best about 1 micron high, and they give the cells what's called the brush border, a fuzzy appearance that is really only apparent when you're looking under a microscope. After each villi, if you see here, this is a villi, note that there's this little dip down after it. That's called an intestinal crypt, or a crypt of Lieberkühn. These are pores that open into glands on the floor of the small intestine that are in between villi. So, like, you would have a villi that goes up, a crypt that goes down, and then another villi that comes up after it. So you're going to find these little pits in between the villi. They are similar in their nature to gastric glands. They have goblet cells and enterocytes to absorb nutrients and secrete mucus in the upper portion. And in the lower portion, they're mostly stem cells. The intestine, like the stomach, is an abrasive environment. You've got food moving through here, you've got digestive enzymes, you've got all sorts of junk going on. That means that cells in the intestines don't last very long. And so this is where the stem cells are hidden and protected, and they are multiplying at a constant rate, pushing more and more cells out to rejuvenate the epithelium of the intestines. There are also a few small cells at the bottom of each crypt called Paneth cells. And these secrete defensive enzymes. Lysozyme, which we've talked about before, but this is going to destroy bacterial cell walls and help to protect against bacteria. Phospholipase, which is going to destroy phospholipids in the cell membrane, particularly of bacteria. And defensins. These are proteins that poke holes in the cell walls, again, mostly of bacteria. These get mixed in with the mucus and help to protect the intestines from attack by mucus or by bacteria. Things moving through the intestines, right? So when things move out the stomach, let's start here. We'll go ahead and use yellow for the stomach, right? You've got food being mixed around in the stomach, and it settles in the pylorus. When the pylorus becomes full, the ileo or the pyloric sphincter opens up, and that chyme is allowed to move into the duodenum. Once you get into the intestines, there are two types of movement that take place. This intestinal movement has three functions. First, it functions to mix the chyme with intestinal juice, bile, and pancreatic juice. Remember that you're going to have the pancreas squirting stuff in here, and just like you needed the stomach to churn in order to mix your bolus with the stomach acid and pepsin, you're going to need to mix that around. You got to knead the dough. Second, to churn the chyme and bring it into contact with the mucosa. Remember that things are only absorbed along the edges. You wouldn't want food to just travel straight through, because then all the nutrients along the edges would be absorbed, but the nutrients that are in the middle of the chyme moving through would never come in contact with the surface, so you've got to mix it up so that all of the various chyme is going to come in contact with the surface. Third, to move residue through. Like, you've got to move stuff along if you're going to eventually pass it out. Peristalsis is one form of movement, and it is most concerned with point 3 here, moving residue along the intestines. What peristalsis is a circular contraction that moves down in a wave. So what you'll get is like contraction starting here, you'll get it to squeeze down, and then that squeezed down portion will keep moving and moving and moving, pushing the food along as it goes. And it doesn't happen like constantly. A peristaltic wave will begin somewhere, usually in the duodenum, and then it'll move 10 to 70 centimeters. Right? So it'll move like that. And then you'll have another wave that starts here and moves further down, and then another wave that starts here. And so you move things down by one segment at a time, basically. You have successive overlapping waves of contraction that kind of milk the chyme towards the colon. This whole process of moving food through the small intestines takes about 2 hours. The other type of movement is called, or motility, I should say, is called segmentation. With segmentation, what you have is different areas of the intestines that will squeeze shut. Now, they don't move. It isn't like peristalsis, where this section will squeeze shut, and then the squeeze shut section will progress downwards. It will be like this section squeezes shut, and then this section opens up, and this section squeezes shut. And this is like, you've got like a bag of something that you want to mix, and you just squeeze it, 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 squeeze it. You're just constantly forming contractions in different places. This does not move the food down, but this fulfills points 1 and 2. What it's going to do is churn and mix the chyme, having the the pancreatic juices and buffer and bile get mixed in with it, and it's going to mix the chyme so that the stuff that was in the middle of the chyme gets brought towards the edge where it can be absorbed. This happens more commonly than peristalsis. It's sort of happening constantly. And it's actually driven not by your brain, but by pacemaker cells, autorhythmic cells that are embedded in the small intestine. And every so often, one of these pacemaker cells will go off and will cause a local contraction in the area. The rate at which these contractions occur depends on how much nutrients there are and where in the intestines you are. All right, so it's about 12 times per minute in the duodenum, about 8 to 9 times per minute in the ileum. When most of the nutrients have been digested and absorbed, and they're just like you're not getting any more nutrients absorbed, the rate of segmentation will go down. Those pacemakers will fire slower, and then peristalsis begins to take over. So in summary, I want you to know the 3 sections of the small intestine: the duodenum, jejunum, and ileum. You should know what order they occur in and about their relative sizes. The duodenum is just that short first segment. The jejunum is the next 40%, and the ileum is the 40% past that. You should know the functions and distinguishing characteristics of the 3 sections. The duodenum is where pancreatic juices are injected and mixed. The jejunum is where digestion and absorption occurs. The ileum primarily is the movement of material through towards the colon, as well as having lots of immune functionality through those Peyer's patches. You should know what circular rings, villi, microvilli, and crypts are, and the functions that each of them plays. And you should understand the 2 different types of intestinal motility and the different roles that they play in digestion. Segmentation is going to be involved in mixing and churning, peristalsis in moving food through the system.